Good afternoon, everyone. My name is Juan Stephen, and I'm going to give you an insight into better management of fish allergy and how we go about doing it. In today's world, we're seeing people move towards healthier lifestyles and eating choices. As such, there's been a massive increase in consumption of seafood being rich in protein, vitamins, fats, minerals, oils. A consequence of this is that seafood allergy has also been on the rise, affecting up to about 5% of children and 2% of adults across the world. Up to about 90% of fish allergy is caused by a single protein called Parvalbumin, which is a small, very robust, heat-stable protein. It's found in two distinct lineages, alpha and beta. It's found predominantly in muscle tissue of all higher vertebrates, including humans. And the beta form of Parvalbumin has been established as the dominant form of the allergenic form of the protein. So what I look at is, I'll present to you a conundrum quoted by a good friend of mine in the audience. If you're allergic to a peanut, you're allergic to a peanut. If you're allergic to a hen's egg, you're allergic to a hen's egg. But if you're allergic to fish, there's over 32,000 species of fish across the world. And Parvalbumin as a protein varies very massively across all these fish species. So for an example, if you're allergic to salmon, it's quite likely that you're allergic only to salmon and nothing else. There's also the possibility that you're allergic to salmon and maybe a few other fish along with it. So what I look at is trying to identify fish species that have Parvalbumin in them and actually try and figure out which parts, what fish actually have allergenic Parvalbumin. The way I go about doing this is people, when they're allergic, they produce an immune response, and that produces IgE antibodies, which bind to specific parts of the protein that we look at, Parvalbumin. And when we're looking at the binding of IgE, we actually identify these specific regions across the protein that actually tell us and establish that protein is allergenic in a particular way. So I'm actually looking at and detecting Parvalbumin across various fish species that are actually specific to those fish and nothing else, also collectively as a whole. And this, in the broader scope, is towards the better management of fish allergy because we're trying to develop immunotherapeutics and actually be able to identify what kinds of people would react to what kinds of fish across the world depending on what they eat, especially here in Australia. And all of this collectively is also leading to me comparing structurally and molecularly across the different molecules, Parvalbumin molecules across the different fish to actually identify and see what you could eat, what I could eat, and what the rest of the world could eat. Thank you.